Hello, I'm Dr. Mason Fedino, and I'm going to be talking about a manuscript that a bunch of my colleagues and I at the Urban Wildlife Information Network, or UN, had published in Ecological Applications last year in 2020, titled Landscape Scale Differences Among Cities Alter Common Species Responses to Urbanization. So, we live on an urban planet, and there's no place where this is exemplified more than in the cities that we live in. However, and this is important, cities are actually quite different from one another. First off, they're not uniformly distributed across space, and there's lots of really small cities with not many people living in them, and then just a few cities that actually have a lot of people living in them. Yet, when you add up all the people living in those small cities, it actually far outweighs any of the other larger cities combined. Second, those smaller cities actually cover more space than much larger cities like Chicago and New York combined, something like that. Yet, and here's my case for larger cities, like say Chicago where I live, people living in them are actually living much closer together than they would be if they were living in someplace much smaller. And it was this sort of relationship where we've got, some cities with lots of people, more densely packed together, and then some cities with not that many people and relatively less densely packed together that really got me wondering whether or not sort of your average urban mammal like your raccoon or your opossum sort of live differently within those cities, that small city versus the large city. Or in other words, as a city becomes more densely populated, does just the relative occupancy of that species change? And when my colleagues and I were thinking about this question, we first just kind of went to a drawing board and looked at these potential relationships graphically. So say, for example, we have average housing density of the city on the x-axis and the proportion of habitat patches a species occupies on the y-axis, or a species occupancy. First off, we could find that there's just a species that have no relationship with this whatsoever. It looks like scattershot. Second, we could potentially find species that actually have a positive relationship between average housing density of a city and their occupancy. And this may be species like pests that many of us may not like, things like cockroaches, things like bed bugs, etc. Finally, there can of course be a negative relationship here. So as an average housing density of a city gets bigger, a species becomes less common in general. And when we were thinking about this sort of graphically, we thought how cool it would be if we could actually come up with this relationship, if we could estimate this type of pattern so that we can actually make predictions to cities that we may not have actually sampled that have a different housing density. But it's not just that. It's not just how common a species is in a city. We are also really interested in whether or not the distribution of a species within that city also changes. So say, for example, is a species like the raccoon more likely to be located in habitat patches away from dense human populations? Or are they most likely to be in habitat patches very near dense human populations? Because these things could actually vary among cities as well. Not only that, does the strength and potentially the direction of this relationship change based on differences between cities? So to get at this question, a number of my colleagues and I realized that we needed to actually be collecting data across multiple cities to address this. And so in about 2016, my coworkers and I at the Lincoln Park Zoo helped start the Urban Wildlife Information Network, which is a systematic multi-city collaboration to understand broad patterns of urban wildlife distributions, both within and between cities. This figure is, of course, just a little old. We're actually up to about 35 cities now. And the way that UN works is that we have a common and systematic study design across cities. At this point right now, we're just, first off, just using motion triggered camera traps. And we're sampling wildlife along an urbanization gradient within each city. So from the core of a city out to sort of the ex-urban or rural parts of a city. And Our camera traps are, for the most part, located in urban green space. Sometimes they're in private land as well. But say, for example, in Chicago and in most other of these cities, they're placed in golf courses, they're placed in city parks, they're placed in natural areas like forests, and they're also placed in cemeteries. And for our gradient design, these are just two sort of examples from Chicago, Illinois, one of our largest cities in terms of population density. And then here's Manhattan, Kansas, actually, with their transects. Those little white dots are where cameras are located. And that's one of the smallest cities that are actually being sampled in this study. So to explore how differences among cities actually have an influence on the distribution of a species within a city, we compiled data from 10 UN partners, some of our 10 oldest partners in this network, from Austin, Texas, all the way to Wilmington, Delaware. I'm not going to read this whole list. You can, of course, pause this video and read them at your leisure. Most of these data came from the summer of 2017, and we analyzed data from 8 species, which are sort of your common urban adapted mammals throughout most of North America. And this includes your coyote, your raccoon, your opossum, your gray and fox squirrel, the cottontail rabbit, the red fox, and the striped skunk. So using occupancy models, which are a statistical tool that can estimate where a species is located as a function of covariates that corrects for imperfect detection, we estimated each species distribution then as a function of one within city factor. So this is something that is calculated at every single location we set a camera trap across these 10 cities. And that was housing density within a 1 kilometer buffer at 472 sites. And then we also calculated 2 between city factors. And this is the really important part of this study because this is the type of covariate that we can only really use when we actually have data from a bunch of different cities. So 2 factors that we calculated here are first, just the average housing density across all of those sampled locations within a given city. So sort of associated to that first one, it's the average within a city. And then that second one that we calculated was the proportion of available habitat within a city, and that was done via NLCD. You can go through the details basically in the paper of how we do this. Dr. Travis Gallo did that part of the analysis. We fit a total of 4 models to each species data. I'm only going to explain the global model because the rest of them are just simplified versions of this model. This global model has a total of 5 different covariates in it. And let me explain what each of these components actually mean. First off, there's these 3 values right here. So the model intercept and these 2 among city variables. And these actually help us estimate differences in the average occupancy sort of among cities. So we can quantify if the average occupancy of raccoon, for example, is different between Austin, Texas or Chicago, Illinois. And these 3 covariates within the model help us estimate where a species is most likely to be found within each city. And so this includes our within city variable, our site housing density, but also the statistical interaction of that within city variable with our 2 between city variables. So we can actually see how a species distribution may change within a city as a function of landscape level features at the city scale. And then finally, sort of using LME4 style syntax in R, we've included a random effect by city within the model to account for any other city variation that's not accounted for by these between city variables. And here's some of the results. So what we see here, we've got on the x-axis, we have average housing density of a given city. And then on the y-axis, we have the relative occupancy, or kind of in simpler terms, the proportion of habitat patches within a city that species may occupy. So we've got this best fit line, the regression from our occupancy model. And then also we've got all these different dots, which are estimates for each of the 10 different cities in this analysis. So what we have here, for example, on the very, very far left, our smallest city, Manhattan, Kansas. Raccoon have an average occupancy of just about like 90%. So raccoon are pretty much everywhere within Manhattan, Kansas. Whereas in Chicago, Illinois, it's actually more like 50%. And you can see it's pretty strongly correlated due to the average housing density of a given city. Not all species responded in this way, though. So, for example, the coyote, they occupy just about 37% of habitat patches, regardless of how densely populated a city is. And this figure here shows that it's not just the average housing density of a city that some species respond to. It may also be the amount of habitat that's actually within a city. And here we see on the x-axis, as the amount of habitat or the proportion of habitat within a city goes up, the average occupancy of cottontail rabbit, which is on the y-axis, actually goes down. So say, for example, in Madison, Wisconsin, the average occupancy is about 50%, whereas in Austin, Texas, it's about 25%. And you may think that, well, if there's more habitat, should that actually be good for cottontails? Yes, probably, but it's also really good for a lot of things that eat cottontails. And so it may just be that as the amount of habitat goes up, it's actually worse off for a species that can do so well in sort of little pocket parks and whatnot, small little urban green spaces throughout a city. It's not, however, just how common a species is and how that may change among cities. It's also where a species is actually located within a city that also changes. There's a number of these figures within the manuscript, and so I'm just going to sort of walk you through one of them. This is for the raccoon, so that you can use this sort of knowledge to sort of decipher what's going on with these other species. And so say we've got the x-axis here, which is average housing density. On the y-axis, this is the effect of site level housing density on occupancy. And so you can think of the y-axis as actually the regression coefficient that would be returned to you if you went out to a city that had one housing density versus another. So say, for example, if we were at a city with an average housing density of 600, and we conducted a study on raccoon, and we used housing density as a covariate, we would potentially find a somewhat positive, but kind of, there may be a positive effect, there may not be. The 95% credible interval here overlaps 0. But if we go out to, say, for example, a much more densely populated city, like, say, around 1400, which is around where Chicago, Illinois is at, we actually see that it's a very strong and negative relationship. So this would mean that for, say, raccoon in densely populated cities, they're not found in the really dense urban core, and they're found more on the outside of the city. Whereas in less densely populated parts of the city, sort of this realm over here, we see that raccoon, for example, may have slightly positive to potentially little to no effect or relationship with housing density at the site level. When we combine all of these results across species and sort of look at patterns of species richness, sort of comparing sites that are surrounded by greater than or less than the average amount of housing density within a city, a really interesting pattern to me sort of emerges here. We can see that actually as sort of a city increases in housing density, we see that the relationship between species richness and habitat patches surrounded by less than average housing density, say like these dots here, and sites surrounded by greater than average housing density here, all of those relationships, or that relationship is negative. So there's fewer species and habitat patches in the urban core of these densely populated cities. Whereas if we look at all the other cities that are less populated, we don't actually see a relationship whatsoever. It goes up, it goes down, sometimes it doesn't change. The big takeaway to me is illustrated in figures like this within the manuscript, because it helps us illustrate how the context of a city is going to have a very strong influence on the relationships and the direction of effect that you can actually find. And so in order for us to actually find similarity among studies, we need to be doing a much better job, talking about the environmental context, the landscape context that a city actually exists within. And of course, we can also use this model to make predictions for new cities that join the network. This, for example, is just the probability of occupancy in sort of an average US city across our analysis. And making these predictions is actually kind of important because while we had 10 cities when we started this analysis in UN, we're now up to 35 different cities. And so we can actually generate testable predictions for partners as they join the network, which is really, really cool in my opinion. And with that, I'd really just want to acknowledge all the partners that graciously provided data for this analysis. And if you have any questions about this, you can reach out to me on my Twitter at Mason Fedino, or you can just sort of look at the UN website at urbanwildlifeinfo.org. Thanks.